On the Evolution of Bacterial Multicellularity, written by Nicholas A. Lyons and Roberto Coulter. Narrated, abridged, and illustrated by Don Inez. Abstract. Multicellularity is one of the most prevalent evolutionary innovations in the biological world. Evidence of single cells independently evolving into multicellular organisms has been found across all three biological domains in many different forms, functioning under almost as many different mechanisms. Scientists have utilized the experimental accessibility and diverse genomic data available in bacteria to study the evolution of multicellularity. This has functioned to reveal the likely selective pressures, possible origins, and even the argued inevitable eruption of multicellularity in all life.
The universe can be viewed in levels of organization based loosely on size. Inorganically, this includes the organization of different subatomic particles into atoms, which themselves can combine in various ways to form molecules. These molecules often exhibit unpredictable properties distinct from their component parts. Eventually, molecules organically shift to macromolecules, and from macromolecules to cells, from cells to multicellular organisms, and from multicellular organisms to ecosystems. Thus, the evolution of life has had a place at each leap of organization, from physics to chemistry to biology and beyond. Of all the transitions between levels of organization, the advent of multicellularity is perhaps the most interesting for several reasons. Multicellularity completely redefines the concept of what an individual organism is, and has erupted independently dozens of times across every domain of life. Multicellularity also allows a transition from the microscopic to the macroscopic world. It inherently causes organisms to be differentially affected by physical laws like Brownian motion and gravity, which is reminiscent of the change from the quantum mechanically driven atomic world to the chemistry-driven cellular world. Multicellularity also enables vast phenotypic expansion and diversification. This results in the advent of cellular differentiation and the temporal development of structures in organisms. Finally, multicellularity is almost definitionally necessary in the formation of intelligence and consciousness as we know it. Like the attempt to define life, defining multicellularity is a tenuous endeavor. However, the scientific consensus is that there are two basic factors necessary in order for an organism to be considered multicellular: cell-to-cell adhesion that forms a new evolutionary unit, and intercellular communication that leads to coordinated activity. Adhesion is used as a loose term to indicate conglomeration by any means, such as aggregation within an extracellular matrix, filamentation by incomplete cell division, or direct contact mediated by transmembrane proteins. Many examples of species and life cycles put this definition of multicellularity to the test. Chiefly among these borderline cases is the widespread expression of multicellularity in the bacterial domain. Long considered the archetypal unicellular organisms, bacteria have become popular as ideal model systems in which to explore the phenomenon of multicellularity, their simplicity allowing them to be well suited to laboratory study. Although multicellular evolution in bacteria is not necessarily the same as that in the other two domains, studying these examples and alternative origins allows us to get to the core of what enables fundamental evolutionary advancements, similar to how studying communication in dolphins can help to reveal the fundamental nature of language. Therefore, bacteria can indicate the selective forces that led to multicellular beings, the genetic underpinnings in their evolutionary history, and why this particular leap in organizational level is so recurrent. The key to understanding how multicellularity evolved is in pinpointing the selective pressures that led to its fixation in so many species in the first place. There are inherent disadvantages to operating as a collective group rather than a single cell. These include the cost of adhesion and communication, the physical limitations of individual movement, as well as the less tangible vulnerability of a cooperative system being exploited by those who do not contribute their share. The fact that conditional multicellularity is so common may be a response to these costs, analogous to how many biosynthetic gene clusters are only expressed when cells are met with certain stimuli. Even with all these costs, the advantages of multicellularity are numerous. It allows for new resistance to physical and chemical stresses, an improved capability to acquire resources, protection from predation, a more efficient mechanism to colonize new territories, increased chances of survival in intermicrobial conflicts, and the opportunity for cell differentiation and specialization. Resistance to environmental stresses appears to be the most common advantage enjoyed by multicellular bacteria. They are more adaptable to varied pH, osmotic pressure, oxidation, desiccation, metal toxicity, and mechanical forces. The evolution of multicellularity as a stress response is an intriguing idea, as that places it in good and evolutionarily significant company. For example, meiosis in eukaryotes may have originally been a response to adverse conditions or accidental increases in ploidy. Likewise, recombination enzymes that evolved to repair damaged DNA were co-opted to share DNA and shuffle genes within a population. This conjugation of DNA greatly accelerated the rate of evolution in bacteria. Multicellular organisms are also better at nutrient utilization. As studies have shown, swarming bacterial predators are more capable of capturing and consuming prey than their non-swarming mutant counterparts. As some organisms have evolved to become better predators, some have also evolved to become less attractive prey. Many studies have found filamentous bacteria to be better at resisting both engulfment and the metabolization of its parts when consumed. Demonstrating this further, co-culturing prey bacteria with predatory protists selects for longer bacterial filaments. This appears to be a regulated response to the protist threat, as bacteria separated from the co-culture by a permeable membrane also increase in length. In addition, the Actinobacterium Streptomyces colicolor conditionally undergoes multicellular development upon exposure to the predatory Mixoxys xanthus. The last advantage of multicellularity discussed here, cell specialization, is one of the most compelling reasons this is such an interesting phenomenon to study. Division of labor, whether within an organism or society, leads to more efficient production and utilization of resources. The classic example of this comes from the filaments of the Cyanobacterium anabaeana, which can carry out both photosynthesis and nitrogen fixation. Due to the chemical incompatibility of these two processes, however, they must be separated into cells specialized for each purpose.